Okay, so the topic of this video is going to be neurons and the nervous system, and we're mainly going to focus on how neurons send messages or signals to other neurons. So let's get started. So when we look at the nervous system, like any body system, the digestive system or the circulatory system, the nervous system is made up of a collection of organs, which are made up of tissue, which are made up of cells. It just so happens that the nervous system helps to control thoughts, movements, and life processes that take place within the body. Now, one thing I want to mention is that the nervous system responds fairly quickly to what we call a stimulus. Light is an example of a stimulus. When you shine light into somebody's eye, like you see in the picture, the pupil gets really, really small. It's trying to prevent too much light from entering the eye. But in dim light, you can see here's an eye in dim light. You can see the pupil, or the black spot in the middle, is much more open to try to let what little light is available into the eye to help you see. But the nervous system responds quickly, but not instantly. If you were to stare into somebody's eyes and turn the lights on, a split moment later you would see the pupil change shape. And so this is something you can try at home just to see how quick the nervous system does respond. When you also think about what must happen in order for this person to pick up the red apple that's sitting on the bench. Well, first of all, you know that light is made up of Roy G. Biv, red, orange, yellow, green, blue, indigo, violet. So white light strikes the apple and red light reflects into the person's eyes. Well, the eyes contain what are called photoreceptors, or rods and cones, and that's going to send a signal. Those rods and cone cells, those photoreceptors in your eyes, are going to send an electrical signal to your brain. That your brain will then receive that signal and start to send another signal to your hand. Now, what we're going to focus on in a few moments is what is the signal that it sends. Well, your brain is eventually going to tell your hand to reach out and grab the apple. So the signal travels down the spine, the signal travels down your arm, and eventually to your hand. And the muscles of your hand then react to this signal and start to reach for the apple. Well, let's look at neurons. Neurons are also known as nerve cells. And the function of neurons or nerve cells is to transfer these signals to and from the brain. These signals are known as impulses. And so as we get through this, we're going to see that neurons are made from three basic parts. The cell body, identified in the diagram, this is what contains the nucleus and the majority of the organelles, such as mitochondria, vacuoles, lysosomes, smooth and rough ER, Golgi bodies. And coming off of the cell body, like in the picture here, you can see there's several dozen what are called dendrites, and they're little branches. And the function of dendrites is to receive these signals or these impulses from a neighboring neuron. So in my animation, that flashing yellow spiky object, that's the electrical signal. And so notice how neurons are in close proximity to other neurons. And so focus on the cell in the middle. The dendrites on the cell in the middle just received an impulse from the neighboring cell. And ultimately, this flashing yellow spiky object, this is symbolic of our signal here, the impulse. It's going to travel down what is called the axon, that long straightaway that you kind of see in the neuron pictured here. Well, what happens when the electrical signal gets to the axon's end? Well, we're going to see that in just a little bit. What I want to next mention are the various types of neurons. First of all, there are what are called sensory neurons. And like the name implies, these are related to our senses. They detect stimuli. And these are the neurons that transmit signals to and from the brain. So in our animation here, a woman's hand is coming in contact with a hot flame. Well, on her hand are temperature signals, thermoreceptors they're called, and their job is to detect heat in this case, and send a signal to the woman's brain, eventually telling her to pull her hand away. And so our sensory neurons are directly related to our five senses, the sense of sight, the sense of smell, sense of hearing, our sense of taste, and our sense of touch. Well, we also have what are a second type of neurons called interneurons, and the reason for the picture is because these are located inside of the brain. Your sensory neurons, which we just previously mentioned, will detect, hot and cold, for instance, and they will send a signal to the brain. So the interneurons actually receive these electrical signals from the sensory neurons. So if you think of, detecting the woman who was had her hand over the candle just a moment ago, she has sensory neurons in her hand that will send a signal to the interneurons in her brain. And then the interneurons in her brain will receive the message and eventually send another signal back to her hand saying to pull away from the heat. And then there are what are called the motor neurons. And motor neurons, like you see in this animation, are going to pass the signal. Again, the signal is called an impulse to other tissue. In this case, the motor neuron is passing the electrical signal to muscle cells. Muscle cells are connected to neurons, to nerves. And when the electrical signal, the impulse, when it comes in contact with the muscle cells, it causes the muscles to react by contracting and stretching. So if you think of the woman again touching the hot candle a moment ago, her sensory neurons touched the hot candle and sent a message to her interneurons. The interneurons will receive that message and send another signal to her motor neurons, and eventually the motor neurons will pull the hand away from the hot candle. Well, now I want to focus specifically on how do neurons send these signals. Well, before we get into that, we need to look at kind of the environment of a neuron. When a neuron is at rest and not sending a signal or an impulse, it's in the state known as resting potential. Well, let's zoom on into the cell membrane of this neuron for a closer look. When we do, we see the exterior of the neuron on top and the interior of the neuron on the bottom. And an electrode, we can actually insert a little tiny electrode into the cell to kind of measure the electrical charge. And what we've learned by doing this is that the interior of a neuron has a negative charge, because inside of the neuron, yes, there's cytoplasm, but the interior of a neuron is filled with proteins that the cell needs in its daily activities. Well, proteins typically have a negative charge to them, and so that's the reason why the internal charge of a neuron is negative, even though I just had some K pluses appear in my animation. The K plus is representative of potassium ions that are also floating around in the interior. But the reason the inside of the cell has a negative charge is just that the positive potassiums are just sheerly outnumbered. There's just a lot more of the negative proteins on the inside of the neuron. So the interior of a cell is measured in millivolts, and it comes in around a negative 70 millivolt. Well, we've also measured the exterior using these electrodes, and what we've learned is that the exterior has a positive charge to it, because outside of the neuron, outside of the cell membrane of the neuron, we see a whole bunch of sodium ions. Na is the symbol for sodium. And so when we look at the resting potential, or we look at a neuron that's not sending an impulse, when a neuron is at rest. When a neuron is at rest, this is kind of the environment that we see. The interior has a negative charge, even though there are some positive potassiums drifting around inside the neuron. The exterior has a positive charge because of all the sodiums. Now, we're going to be focusing our attention a lot on the sodiums and the potassiums. In a few moments, I'm going to remove the negative proteins just because they're going to get in the way, and they're just kind of added clutter to the animations that follow. But just keep in mind that they're still there. So when a neuron is at rest, it has what's called resting potential. But let's say you touch something hot with your hand. Well, then the neuron will send a signal, and this is known as an action potential. Well, the analogy I like to make is that an action potential is like people doing the wave at a sporting event. One person stands up and sits down. Then the next person stands up and sits down. Then the next person stands up and sits down. And I hope you'll see why I chose this as a nice analogy as we move on. But the definition of an action potential, an action potential is that impulse, is that electrical signal that we've been talking about. And that electrical signal or the impulse will move down the length of a neuron, down that long straightaway known as the axon. And so let's talk about how a neuron generates the impulse. In my animation, I have the inside of the cell. Just remind yourself that there's a negative charge, even though I did not draw all the negative proteins that are typically found inside the cell. Just to reduce clutter, I removed the negative proteins that are found inside the cell, and I want you to focus your attention on the positive sodiums and the positive potassiums. So the interior cell, again, has a negative charge, and around negative 40 millivolts. Watch what happens. Around negative 40 millivolts, those black sodium doorways, black sodium channels are going to open, and that's going to allow sodium to diffuse into the cell, bringing the normal negative interior up to a positive charge. Because again, when all that sodium rushes in because of diffusion, sodium has a positive charge, and it brings the charge up from the negative range into the positive range. And that's what an impulse is. An impulse is not a physical object that you can hold in your hand. An impulse is that change from negative to positive. Well, what happens next is once the charge reaches a certain strength of positivity, plus 50 millivolts as you see in the animation, it causes the red potassium doors to open, the red potassium channels to open, and then the potassium will simply diffuse out of the cell, dropping the charge. Again, you got to keep in mind that when you lose a bunch of positive potassiums, when you lose a bunch of positive charges, the charge is going to go back down into the negative range. And so again, this is what an action potential is. It's the change from negative to positive back to negative. And eventually the area returns again back to its negative charge. And this process continues down the entire length of an axon. And I hope you're going to see in just a moment why I think the analogy of people doing the wave at a sporting event works well. Let's look at this next animation. So in this case, here's an action potential or an impulse moving down a neuron. And as it moves down the axon right now, we're going to freeze it right here. Let's zoom on in and get a closer look. So again, here we have the sodium on the outside, the Na, and the K or the potassium on the inside. Now, keep in mind that normally the inside of the neuron has a negative charge. I simply did not draw all the negative proteins inside the neuron because I just didn't want to add to the clutter. I want you to focus on the sodium and the potassium. But just remember that inside of a neuron, there's a negative charge. And so what happens is the black sodium channels are going to open, and that will cause the sodium to diffuse in. That brings this normally negative charged area into the positive range. That's the impulse. And when the positive charge becomes strong enough, it causes the black sodium doors to shut, and it causes the red potassium doors to open. And once that happens, the K or the potassium can exit the cell, dropping the charge back into the negative range. Once the charge becomes a certain negative strength, those red potassium doors will shut, and the next set of black sodium doors will open, and the process repeats. It's kind of like a person at the sporting event doing a wave. Person 1 stands up and sits down, and then person 2 stands up and sits down. So now that the sodium channels have opened, the sodium again will rush in, making this normally negative area positive in charge. When it becomes a certain positive strength, the black sodium doors will shut and the red potassium doors will open. That allows the potassium to exit and the charge drops back down into the negative range. It's like, again, a person doing the wave, standing up and sitting down. When the charge reaches a certain negative strength, the the red potassium doors shut and the next black sodium doors open. And that causes, again, the sodium to diffuse in, bringing the charge up to the positive range. Once it becomes a certain strength of positivity, the black sodium channels shut and the red potassium doors open. That causes the potassium to exit, again dropping the charge back into the negative range. Here's another animation that kind of gives you a simpler view, but also I think it really shows what we mean by like the analogy of people doing a wave. As the action potential moves down, the charge spikes into the positive range and then drops back to negative, almost like a person standing up and sitting down at a sporting event. So every time the sodium rushes in, the charge becomes positive, and every time the potassium exits, it goes back to being negative. So after the impulse is passed, you're kind of left with a big mess. And now there's a problem. Now you've got too much sodium on the inside of the cell and it doesn't belong there. It belongs on the outside of the cell. And we also have too much potassium on the outside of the cell and it normally is found on the inside of the cell. So something has to reset the cell back to its normal environment. And that something that's going to clean up the mess is known as the sodium-potassium pump. So who cleans up the mess is what is called the sodium-potassium pump. And how does it happen? Well, through the process of active transport, the use of ATP. In my animation, I have the black sodium, 1, 2, 3 sodium ions at a time will enter the sodium-potassium pump. And with the help of ATP, when ATP is broken, heat is released. That heat causes the sodium-potassium pump to change shapes and therefore pump out the sodium that originally diffused into the cell. Well, it's called the sodium-potassium pump because potassium, 1, 2 potassium ions at a time, are then pumped back into the cell. So when does this happen? This happens after the impulse has passed. Where is the sodium-potassium pump located? Well, it's located in the cell membrane. But most importantly, why is this relevant? Well, this has to happen in order to restore the cell back to its normal resting potential. That way, the cell can be ready to send another impulse in the future if needed. So here is our animation as we left it, and now I added the blue sodium-potassium pumps. And again, 3 sodium ions at a time will be released, and 2 potassium ions at a time will be returned. Now, if you imagine all the sodium-potassium pumps working at the same time, the cell can be returned to its normal condition in quite some time. So imagine that all 6 of the blue sodium-potassium pumps just got done doing their job. There you go. The cell will be back to its normal negative condition in a relatively short amount of time. So now that we have zoomed back out to these 2 neurons, let's continue the movement of the impulse or the action potential. And now that the impulse has reached the end of the neuron on the left, let's zoom on in for a closer look. Now, what we're seeing here is the little gap in between the two neurons known as the synapse. There's a real small microscopic gap in between the neuron on the left and the neuron on the right. And this small little gap here presents us with a problem. Well, let's see. Sodium comes in and potassium comes out. So we see the impulse again is moving. Sodium comes in and potassium comes out. And one more time, sodium comes in. There's the impulse again. And now notice how the impulse has nowhere left to go. It's reached the end of the neuron on the left. And this synapse, this gap, represents a problem because the impulse can't cross this gap. Again, the impulse is just this change of positive and negative electrical charges. But there is a solution. Neurons contain proteins by the name of neurotransmitters. The cell on the left in a moment is going to release neurotransmitter proteins to the cell on the right. And so what we see in this process here is that the impulse has reached the end or the terminal of the cell on the left, the neuron on the left. So the impulse has nowhere left to go. It's reached the terminal end. Well, what happens next is you can see that there are 3 vesicles in the cell to the left. And a vesicle has just fused with the cell membrane and is slowly releasing the black neurotransmitters, which simply diffuse across that gap across the synapse. And notice how one of them landed on a receptor to the neuron on the right. Okay, so the receptor will catch these neurotransmitter proteins. And what it kind of happens next is pretty neat. When it catches the neurotransmitters, that will so now that the receptor on the neuron to the right just captured a neurotransmitter, that will stimulate the first round of sodium channels to open. Once that happens, sodium diffuses in. That brings the charge up to the positive range. A new impulse has been created. And eventually, the potassium channel will open and potassium will exit. That will drop the charge into the negative range. That causes the next sodium channel to open, and then sodium will diffuse in, bringing the charge up into the positive range. That will cause the red potassium channel to open, and potassium exits. It's a new action potential. It's a new impulse. And so the process simply continues like we said a moment ago. Sodium rushes in, it creates a positive charge in an area that is normally negative, and that will cause the potassium doorways to open so potassium can exit. The next round of sodium channels will open and sodium will diffuse in, creating the positive charge. That will cause the next set of potassium doors to open and potassium to exit. And then that will cause the next round of sodium channels to open, creating an impulse again, which will then cause the potassium channels to open and the potassium exits. So again, what we saw here is how an impulse can be triggered in a brand new cell. And again, now we're coming to the end of the second neuron here. So the impulse reaches the terminal end, sodium rushes in, the charge becomes positive, potassium exits. Sodium rushes in, the charge becomes positive, potassium exits. Sodium rushes in, the charge becomes positive, and now notice how again the impulse has nowhere left to go. So the cell on the left will now release its supply of neurotransmitters. So the vesicle will fuse with the cell membrane and slowly release neurotransmitters. The neurotransmitters attach themselves to a receptor on the cell on the right that stimulates the creation of a new impulse. A sodium channel opens, sodium rushes in, potassium exits. And then that causes the next sodium channel to open, sodium rushes in, and that causes the potassium channels to open, allowing potassium to exit. And so let's say you want to kick a soccer ball. You're looking at the soccer ball with your eyes, and your brain is about to tell your feet to kick a soccer ball. Well, how does this work? Well, your brain will send an impulse down one neuron, which will release neurotransmitters to the next neuron. The second neuron will receive the neurotransmitters and create its own impulse. The impulse reaches the end of cell number 2. Cell number 2 will release its own neurotransmitters, which will create an impulse for cell number 3. Cell number 3, now that the impulse has reached the end of cell number 3 will release its own neurotransmitters, and cell number 4 will create an impulse. The impulse reaches the end of cell number 4, releases neurotransmitters, which arrive at cell number 5. Now, in reality, this process, of course, happens much, much faster. We're just slowing it down to talk through it. And eventually, the impulse reaches the muscles of your foot. And when the impulse, when the electrical charge comes in contact with muscles, the electrical charge causes the muscles of the foot to stretch and contract in a very coordinated manner, allowing you to hopefully successfully strike the soccer ball and perhaps score a goal. Now, keep in mind, when we're young, we're not very coordinated at our muscle movements. But as we age and just more experience, we become much more coordinated. And so there you go. There's the end of these notes. I hope you see why I think an action potential is kind of like people doing a wave. One person stands and sits, kind of like the charge going into the positive range and then into the negative range. So go ahead and pause this video. If you're in my biology class, try to answer these questions, and I'd be happy to check your answers before school or after school. Good luck.